So, in the week one of host pathogen interactions, we discussed a little bit about the introduction, terminologies, then Alimechnikov, then Emil von Behring, then about toxin neutralization by Ehrlich, then the Vidal test, the Rem Vidal test, Hans Ernst August Buchner, then Pfeiffer phenomena, then Jenner, then theories of immunity, then immune systems, regulation of immune responses. So these were all that was discussed, and let's see the questions of the assignments that was given. So the first question is, which experimental approach proved that serum from animals exposed to toxins could protect other animals? So we discussed regarding this, which is that whether it is phagocytosis study by Mechnikov, of serum therapy experiments by Behring, or the toxin neutralization by Ehrlich, discovery of complement by Bordet, agglutination studies by Gruber-Durham. So we discussed which one is that proved that serum from animals exposed to toxins. So you can answer this. Then next we also discussed the Gruber-Vidal test. So what is the primary purpose of Gruber-Vidal test? Whether it is to detect the antibodies against Salmonella typhi in the patient's serum, or to diagnose the typhoid fever, or to identify the bacterial toxins in the blood for rapid typhoid diagnosis, or to observe bacterial growth patterns in blood samples, or to differentiate between typhoid and other bacterial infections using heat stable antibodies, or whether it is to detect complement activation leading to bacterial lysis in typhoid patients. So we discussed the Gruber-Vidal test in detail, and which one of these is the primary purpose is known. The next question is, which of the following statements best reflects Buckner's 1889 discovery regarding the alexin in blood serum? So we discussed Buckner's discovery of alexin. So whether Buckner identified alexin as a precursor to antibodies, or highlighting its role in adaptive immunity, or Buckner demonstrated that alexin could destroy bacteria independently of immune system, or whether Buchner proved that alexin requires the presence of leukocytes to function effectively against bacterial pathogens, or Buchner discovered that alexin selectively targets gram-positive bacteria, ignoring the gram-negative strains, or whether Buchner concluded that alexin is a heat-stable protein responsible for bacterial destruction. So we discovered what all the points he told us. And which of the following concepts have contributed to the foundational theories of immunology? So we discussed the foundational theories of immunology and what were the key concepts there. Whether there are multiple answers here, so you can select all of them. Mechnikov phagocytic theory, Ehrlich's side chain theory, or evolutionary theory, or the complement system, or Behring's serum theory, therapy, or whether it was the theory of gene expression, the dichotomy between cellularists and humoralists. We discussed this also, and the study of monoclonal antibodies as well. So you can select all the correct answers over here. And next is, what is the key observation in the Pfeiffer phenomena? So whether it was Vibrio undergoes phagocytosis in immune animals, or Vibrios are killed by immune cells in the peritoneal cavity, or Vibrios undergo bacteriolysis in immune animals, swelling, dissolving, and disappearing within 20 minutes, or immune serum prevents Vibrios from multiplying in the test tubes. Vibrios are engulfed and destroyed by cellular activity in immune animals. We discussed the key observations of Pfeiffer's phenomena. So you can choose the correct answer here. Then, what did Edward Jenner use to develop the first successful vaccine? So we discussed the Edward Jenner's first successful vaccine, which virus he had taken, and against what virus, and how the process was done. So whether it was smallpox, cowpox, rabies, influenza, or polio. So you should remember against which particular virus the vaccine was developed, even though it was taken from a similar type of virus to use. Then, what was the main difference between the cellular and the humoral theories of immunity? So we discussed the cellular and the humoral theories of immunity. So the cellular theory emphasizes immune cells, while humoral theory focuses on soluble factors in the blood. Cellular theory highlights antibodies, while humoral theory focuses phagocytosis. Which one of these are correct? Cellular theory concentrates on complement proteins, while humoral theory does not. Cellular theory is applied to bacterial diseases, while humoral theory is applied only to viral infection, or cellular theory ignored the role of macrophages, while humoral theory emphasized their importance. So, which one of these options are correct regarding cellular and humoral theories of immunity? Next is which type of immune specificity is primarily associated with innate immunity? So, whether it's narrow specificity for unique molecular structures, or broad specificity for molecular patterns, or memory specificity for prior infections, or tolerance specificity to avoid self reactivity, or adaptive specificity that develops over time, which among these is the correct answer? Which of the following contributes to regulation of immune responses? So there are multiple answers for this. So among these, which ones are correct? Whether pro-inflammatory and anti-inflammatory cytokines, or pattern recognition receptors in adaptive immunity, or positive negative feedback mechanism, or cellular interactions such as M1, M2 macrophage balance, complement activation pathways, or the role of bacteria in vaccine development, antigen specific antibody production, or phagocytosis of the pathogens. Select all the correct answers. Then, whose work led to the development of first vaccine? Whether it was Louis Pasteur, Robert Koch, Edward Jenner, Thucydides, or Rudolph Virchow. So we discussed this as well. So let's see. Yeah, so we discussed all the topics. Yeah. So based on this, you can answer the questions. So if there are anything else, you can mail me to this email ID.